A pleasant day to everyone. We are Group 3 of Grade 12 STEM 2301 and will be presenting our research entitled as Formulation of Diabetic-Friendly Jelly Bears using guava for blood sugar level control. Throughout this presentation, we will introduce you and guide you through the background of our study, the methods we employed, our key findings, and its conclusions and implications. Diabetes is a condition characterized by high blood sugar levels, which, if left uncontrolled, can lead to serious complications. One of the biggest challenges for individuals with diabetes is finding snacks that don't cause harmful spikes in blood sugar. Many common snacks are high in sugar, making it difficult to keep blood glucose levels stable. This is where guava comes in. Guava is rich in fiber and antioxidants and has a low glycemic index, which makes it an ideal fruit for regulating blood sugar. Studies have shown that guava can help prevent blood sugar spikes, which is especially helpful for managing type 2 diabetes. Our research aims to formulate diabetic-friendly jelly bears using guava. These jelly bears will serve as a healthy and enjoyable snack option for people with diabetes, offering a delicious alternative without compromising blood sugar control. By incorporating guava, we hope to provide a nutritious, safe snack that will help individuals with diabetes better manage their condition while enjoying a tasty treat. The purpose of this study is to develop formulations of diabetic-friendly jelly bears using guava, with the aim of providing a healthier snack option for diabetic individuals. This research seeks to evaluate the glucose release of these jelly bears through in vitro digestion method and God pod method. With this study, we will create a health-conscious snack alternative that promotes better blood sugar level control for people with diabetes. With that, our research seeks to answer the following questions. Question number 1. What is the average glucose level concentration of the different formulation of the guava jelly bears? Question number 2. What is the rate of glucose release between different formulations of the guava jelly bears in terms of oral phase, gastric phase, and intestinal phase? And lastly, question number 3. What is the pH range of the different formulation of the guava jelly bears?
For the significance of the study, first is the diabetic individuals. This study introduces diabetic-friendly guava jelly bears as a snack option that helps regulate blood sugar levels. It provides individuals with diabetes a healthier alternative that fits their dietary needs and support blood sugar control. Next are the researchers. The findings of this study offer a foundation for creating innovative snacks for people with diabetes. It highlights the potential of guava in managing blood sugar, inspiring future or further research and product development in diabetes management. For the last one are the future researchers. This study serves as a guide for future research, detailing the creation of diabetic-friendly jelly bears and encouraging further exploration of natural ingredients for healthier and functional foods.
Scope and delimitation. This study aims to formulate diabetic-friendly jelly bears. The goal is to control blood sugar level through the consumption of these jelly bears. The formulation utilized guava as the main ingredient and agar-agar as the sole gelatin substitute, ensuring it is suitable for individuals with dietary restrictions. The research focused on determining the glucose release rate of the formulated guava jelly bears during each phase of in vitro digestion, oral, gastric, and intestinal digestion. The glucose release was quantified using the glucose oxidase peroxidase or God pod assay, a reliable biochemical method for measuring glucose concentration. This study is limited to laboratory-based research and did not involve human or animal testing. It was solely focused on producing data for academic purposes. The findings were documented in a research paper and would not extend to clinical trials or commercial applications.
Methodology. Research design. To determine whether the consumption of jelly products developed reduces blood sugar levels, the researchers used a quantitative method using an experimental research design that focuses on the formulation and production of sugar-free vegan jelly bears. The researchers analyzed the digestion process and glucose release using the in vitro digestion method and the glucose oxidized, peroxidized, or God-pod method. These methods are carefully selected to provide precise insights to the formulation's effectiveness on blood sugar control. Researchers chose these methods because it allows the researchers to collect and validate the data needed.
The study was conducted at NU Nazareth School. A total of 10 guava jelly bears were needed for the 3 trials for each of the 3 formulations for the oral, gastric, and intestinal phases of the in vitro digestion, for a total of 27 tests. The dimensions of each jelly bear formula are 1 cm in width and 2 cm in height. The formulation of every phase of guava jelly bear are the following. Phase 1 researching model paper for fruit concentration of the guava and agar. Phase 2 intermediate formulation has been modified based on the findings from Phase 1. And then last, Phase 3 final formulation focus on maximizing both flavor and nutritional benefit. This approach ensures that the guava jelly bears has the potential to help diabetic individuals control their blood sugar levels.
This study will utilize an in vitro digestion model to simulate the human digestive process, specifically evaluating the glucose release from diabetic-friendly jelly bears formulated with guava. The model will replicate the oral, gastric, and intestinal phases of digestion, following established methodologies to ensure precise and accurate assessments of glucose release and bioavailability. The in vitro digestion model is an effective tool to mimic the human digestive system, providing valuable insights into how the jelly bears interact with digestive enzymes, without the need for clinical trials. To measure the glucose levels in the jelly, the glucose oxidase peroxidase or God-pod method will be employed. This technique uses glucose oxidase to convert glucose into gluconic acid and hydrogen peroxide, which is then further broken down by peroxidase to produce oxygen. This method enables the precise quantification of glucose in the digested samples, allowing for a thorough evaluation of glucose bioavailability and potential impacts on blood glucose levels.
pH measurements. Measuring pH is essential for assessing the acidic or alkaline qualities of digested jelly bears, providing insights into their response to stomach acidity and intestinal alkalinity. pH levels play a crucial role in determining both the stability and the release of glucose from the jelly bears, which is particularly important for evaluating their suitability for individuals with diabetes.
To develop guava jelly bears for individuals with type 2 diabetes, we prepared 3 formulations. Formulation A involves mixing 49 grams of guava juice with 49 grams of water, then adding 2 grams of agar, and heating until dissolved. The mixture was poured into molds and allow it to set. For formulation B, it consisted of 45 grams of guava juice, 50 grams of water, and with 2 grams of agar, heated and then refrigerated. Formulation C used 59 grams of guava juice and 46 grams of water with 4 grams of agar, also heated and refrigerated. For the in vitro digestion procedure, we started with the oral phase by crushing 10 jelly bears, measuring their weight, and adding the alpha amyls accordingly. The mixture was stirred at 37 degrees Celsius for 2 minutes. In the gastric phase, pepsin was added, and the mixture was stirred for 2 hours at pH 3, with pH adjustment made with using HCl if necessary. A sample was extracted for glucose analysis. The intestinal phase involved mixing with bile salts and pancreatin, incubating at pH 7 for 2 hours while continuously stirring and monitoring pH. To know glucose release, we used the God-pod method. We prepared blank, standard, and test tubes, adding glucose reagents and appropriate sample to each The calorimeter was calibrated with a blank tube, and the test tube was incubated at 37 degrees Celsius for 10 minutes before measuring glucose release. Finally, to determine the pH of the jelly bears, we disperse 1 gram of the formulation in 100 ml distilled water and measure the pH using the digital pH meter.
So for our data analysis, to validate the findings, a descriptive analysis was conducted to compare glucose release across 3 jelly bears formulation, which is A, B, and C, during the oral, gastric, and intestinal phases of in vitro digestion. Using the GOD-POD or glucose oxidized peroxidase method, glucose release from 10 jelly bears per formulation was quantified, while pH levels were measured from 1 jelly bear in each formulation to monitor changes affecting enzymes activity. The average glucose concentration was also analyzed to assess the formulation's effectiveness in maintaining lower glucose levels, providing insights into their potential for glucose management in diabetic individuals.
As for ethical considerations, this quantitative research on formulating diabetic-friendly jelly bears from guava fruit adhered to strict ethical standards, as it involves no surveys, participants, or testing on living subjects. These studies focus only on laboratory-based experiments. Researchers ensured responsible handling, processing, and disposal of ingredients and chemicals to minimize waste and environmental impact. Proper documentation and transparency were maintained throughout the process.
Results and discussions. This chapter presents and discusses the results of glucose release from guava jelly bears using in vitro digestion simulation and subsequent analysis with a GOD-POD or glucose oxidase peroxidase assay assisted by a calorimeter. The findings highlight the presence or amount of glucose release during each digestive phase, oral, gastric, or intestinal digestion, and with the effectiveness of the GOD-POD method in quantifying these glucose concentrations. The average results shows that Formulation C has the highest initial glucose concentration of 1.95 mg per 100 ml and the highest total calculated average of 390 mg per deciliters. Formulation A follows with 1.73 mg per 100 ml and 346 mg per deciliter, while Formulation B has the lowest values, which is 1.56 mg per 100 ml and 312 mg per deciliters. These differences highlights variation in glucose release among the formulations, which answers our research question number 1.
This research examined the disintegration of 3 guava jelly bear formulations A, B, and C during oral digestion using the GOD-POD technique to measure glucose release. Findings indicate significant differences in glucose levels. Formulation C had the highest at 399 mg per deciliter, followed by A at 346 mg per deciliter and B at 312 mg per deciliter. In the oral phase, C's the glucose levels peak between 397 mg per deciliter and 422 mg per deciliter, while B had the lowest values.
One of the results in our research examined the glucose release of 3 different jelly bear formulations A, B, and C during the gastric phase. Each formulation was tested in 3 trials to determine how much glucose was released. Formulation C had the highest glucose release, ranging from 381 to 388 mg per deciliter, suggesting it contains more sugar or releases glucose faster. Formulation B had the lowest glucose release, between 300 to 315 mg per deciliter, indicating a lower sugar content, while Formulation A was in between, with values ranging from 325 to 338 mg per deciliter, showing moderate glucose release.
Glucose release in gastric phase varied among formulations, with lower values indicating slower digestion and delayed absorption, as stated by Muller et al. 2018. This happens because food stays in the stomach longer before reaching the small intestine, where glucose is absorbed. According to Regid-Heda et al. 2023, pepsin, the main enzyme in this phase, breaks down proteins but does not digest carbohydrates, leading to less glucose release compared to the oral phase.
The intestinal phase showed a more varied pattern than the gastric phase, where Formulation C consistently had the highest glucose release. Formulation C was the highest in all three trials, rising from 1.55 milligrams per 100 milliliters in trial 1 to 1.77 milligrams per 100 milliliters in trial 3. This means that in comparison to other formulations, Formulation C experienced a more thorough breakdown of glucose during intestinal digestion. We are looking at the intestinal phase total glucose concentration. The data show the consistent rise in glucose release, indicating the fact that the digestion continues to break down the jelly bears after the gastric phase. Formulation C consistently had the highest concentration in trial 3. It reached the highest amount ever recorded, 402 milligrams per deciliter. This demonstrates the act of digesting increases the amount of glucose available in the intestines. Continuing, we are looking at Table 4, which provides a summary of the glucose levels in every trial. It shows that Formulation C breaks down the most sugar in the intestines because it had the most or highest glucose release, which is 352 to 402 milligrams per deciliter, where Formula B had the lowest release from 304 to 315 milligrams per deciliter, indicating a slower breakdown of sugar. Formulation A had the moderate levels from 311 to 365 milligrams per deciliter, suggesting a consistent breakdown. The observed patterns indicates that each formula's composition has a significant impact on the availability of glucose. We relate these findings to an external study by Diaz-Pinheiro 2024, which examined the influence of bile salts on digestion. The study discovered that the higher bile salts concentration promote digestion by breaking down food structures. However, they may also interfere with glucose movement, potentially slowing down its absorption. This helps explain why glucose release can vary during the intestinal phase. Understanding the role of bile salts and structural breakdown could provide a valuable insights into how different formulations behave during digestion.
According to Gostock in 2024, the pH value is critical for gelation and gel stabilization since gel-forming agents form gel at isotonic points in confectionery production. The pH of the jelly bears were created with various formulations was found to be between 6.12 and 6.27. These numbers were influenced by varying ingredient percentages. Based on June in 2021, the pH noticeably affects the strength of the agar gel. As the pH decreases, the gel strength weakens. In this manner, as the pH of the jelly bear increases, the gel strength increases also.
Conclusion. The study analyzed the glucose release of 3 jelly bear formulations without enzyme intervention. Formulation C had the highest glucose concentration at 390 mg per deciliter, followed by A at 346 mg per deciliter and B at 312 mg per deciliter. In the oral phase, Formulation C consistently exhibited the highest glucose values, while A and B showed lower concentrations. During the gastric phase, Formulation C remained the highest, with A and B showing reduced glucose release. In the intestinal intestinal phase, C again maintained the highest release, suggesting a higher sugar content or faster breakdown, while A and B it displayed lower values. The pH levels of the formulations range from 6.12 to 6.27, influenced by guava content.
Our study found that modifying glucose concentration and gelling agents can enhance the taste, texture, and structural stability of guava jelly bears. The current formulation exhibits a slightly watery consistency, which may be improved by increasing the concentration of the gelling agent or exploring alternative gelling agents. Furthermore, maintaining controlled glucose release is essential to ensure the product remains suitable for individuals managing blood sugar levels. Positioning the jelly bears as a diabetic-friendly, low-glycemic snack may enhance their appeal to health-conscious consumers. Additionally, collaboration with nutrition experts could provide scientific validation and further support their potential as a healthier alternative to traditional sugary confections.
Future studies should include larger human-based clinical trials to confirm the long-term effects of these guava jelly bears on blood sugar levels, building on the in vitro digestion testing used in this study. These trials should take real-world eating habits into account, as in vitro digestion method is only a lab-based simulation or replication of human digestion. Moreover, testing different varieties of guava could help ensure consistency in glucose release. By expanding the research to include diverse groups of people, future studies could provide more personalized recommendations for managing blood sugar levels, offering insights for individuals with different types of diabetes.
The study highlights the significant limitations due to constrained budget for equipment procurement. Researchers must seek more affordable alternatives to replicate the functionalities of advanced tools, often resorting to low-cost substitutes. However, those options frequently lack the performance and efficiency of their sophisticated counterparts, compromising the quality and reliability of the research findings and limiting the ability to conduct experiments to the desired standard.
As for our appendices, firstly we have Appendix A, Materials and Instruments, and then Appendix B, Oral Phase Documentation, followed by Appendix C, Gastric Phase Documentation, and then Appendix D, Intestinal Phase Documentation. Next is Appendix E, Completion of Paper and Data, and lastly, Appendix F, Informed Consent Letter.
And that concludes our oral presentation. Thank you for listening.